appreciate Bio-Rad, and thank you all for coming today. I really do appreciate people taking time out of their busy schedules. It was a while back when I finally did retire in 2006 for full-time work, but then I did some part-time work at a reference laboratory at LifeShare in Shreveport. And then, crazy me, after not being in a hospital over 40-some years, I decided that I would work at a hospital for a while, and 6 months was enough. I knew exactly what it was like at this type of facility, but I really did enjoy my work at Good Shepherd Hospital. And I still have my little badge, and I can clock in, and I work PRN, and when they have special and interesting cases. But a couple of years ago, the American Red Cross asked me to write a review on LW for the journal Immunohematology. And it was a work in progress for 2 years, I think. First I said no, and then I said yes. But anyway, it brought back all kinds of memories about the first years of LW and the association with RHD, and it was really fun going down memory lane. And that's probably where I'm taking you today, to give you all the stories and the fun stuff about these blood groups, which is one of my first loves. In 1939, Levine and Stetson investigated a case of erythroblastosis fetalis. The maternal antibody reacted with 80% of ABO-compatible individuals, and an interesting fact is that this was the woman's second pregnancy, and she needed a transfusion, so they transfused the blood of her husband. And of course, this was something that was a very common practice in those days, before they knew that that was not the right person to be using for a transfusion. And it was a new system, but they didn't give it a name at that time. And this work was done in New York. And this picture is Philip Levine. And many, many people know the Levine laboratories in ortho clinical diagnostic. The lab was named after Philip Levine, and a wonderful investigator. He really, he was very vocal too, and very much interested in the RH blood group system, the RH system. Then in Landsteiner and Wiener in 1940, who also did work in New York City, they immunized rabbits and guinea pigs with red cells of rhesus monkeys. The antibody reacted with 85% Caucasian red cells, and they named the antigen rhesus. And it appeared to be the same as the human antibody that was described by Levine and Stetson. In 1942, however, Fisk and Foord demonstrated that all cord cells were Rh positive, not 85% or rhesus positive. And you'll notice that I put cord in bold, because this is one of the very first serological tools we had to work with, is cord red cells, when we were dealing with antibodies that either appeared to be anti-D-like or anti-LW in our reference lab. So a very valuable serological tool. Some thought at that time that the animal reagents were not as reliable as the human reagents. So they still were thinking that it was the same antibody, the human and the animal ones. The rhesus blood group was defined by antibodies, and the Rh factor was termed D by Fisher in 1944 when he devised the CDE nomenclature. And a lot of us have a better understanding of RH with this CDE nomenclature than we do with the Rosenfield numbering system or the other nomenclature. So Fisher was the first one, and it was actually 1944 when he did this. Murray and Clark and Levine in 1952 and Levine in 1967 did quite a bit of work and showed that D negative or Rh negative and D positive, Rh positive red cells stimulated an apparent anti-D-like antibody in guinea pigs. Levine confirmed this in 1961 by adsorption studies. Levine in 1967 was the one who renamed the anti-D-like factor, anti-D-like, in LW in honor of Landsteiner and Wiener. And this is the first and only blood group system that I know of that is named after the investigators. Most of the blood group, not all of them, are named after the antibody producers like Colton. Actually, it was interesting because Colton should have been Calton because her name was C-A-L, but somebody's handwriting didn't show up and it was named Colton. So a little bit of story on that. So after the antibody producers, but this is one that's named after the investigators. LW antigens and antibodies. The first two human antibodies to the D-like, or LW antigen, were seen in 1955. They were not reported on, though, until 1962, so don't get discouraged if you find something interesting and it took 10 years or 12 years to get it published. And I don't know how many of you were at the ABB in Baltimore, Maryland, but they had a workshop or a seminar on cold cases, and it was really kind of fun. And to see how it took 20-some years to investigate a particular case and then finally get it published. But the newer tools that we have now can explain a lot of our serology. These were two D positive individuals, G and B. So here they were D positive and they had produced a D-like antibody. Now we do know that today you also can be genetically D positive, have a partial D antigen, and produce allo-anti-D. So that's another consideration when you're looking at whether it's Rh-anti-D or whether it's anti-LW. So all these different aspects can make it kind of, you have to really do a lot of investigating work to see which case you're really dealing with. But it's fun. This is a picture of Jane Swanson on her retirement. Jane Swanson, after World War II, she and two other ladies started War Memorial Blood Bank in Minneapolis, Minnesota. It's now called Memorial Blood Center. And one of the ladies, Eleanor Amberg, was the lab manager of the blood center, and Helen Arndt was the instructor. She taught every new employee, and she also was the instructor for the SBB school. They had an SBB school. But Jane's love was blood groups, and from day one, she lived and breathed blood groups. And actually, it was kind of interesting. I love telling these stories about people, but Jane's mother is YTA negative. And she insisted when her mother went in for surgery that she got in YTA negative blood, even though she didn't have anti-YTA in her serum. So Jane wasn't really that popular with the blood bank staff. But Jane worked on the third example, and VW are the initials for this one. And she and Dr. Matson reported it in 1964. This was a case where the cells of a sister were compatible. This is very important, because if you have a sibling who lacks the antigen, then we didn't have molecular in those days to show whether you were genetically LWA negative or not, then it probably is a case of allo-anti-LW because you have a sibling who lacks the antigen. VW cells were non-reactive with the first two human antibodies and also the guinea pig anti-LW. Levine in 1967 first suggested that anti-LW was composed of fractions, anti-LW1 and anti-LW2, analogous to anti-A and anti-A1. This was, LW1 was for Rh positive cells and LW2 for Rh negative cells, because they indicated and showed in their studies that the LW antigen appeared to be stronger on D positive or Rh positive cells than on Rh negative cells. So this began the first numbering of the LW antibody antigens. Then came along a case of Mrs. Big, and her real real name's Bigelow, and it's in the literature. I understand that you can use names if it's in the literature, so I continue to use the names of these important people. But she had a very potent anti-LW in 1971. The titer against Rh positive or D positive cells was 32,000. The titer against Rh negative or D negative cells was 400. And again, this is kind of showing that this appeared to be like an anti-D-like antibody, but it was anti-LW. Her cells of one of her brothers was compatible, and the eluates of BIG and BW were compatible. Now, you have a situation serologically that occurs where ABO interferes with some of your testing, and one of them was a B and one of them was a group A. So you couldn't directly test them back and forth. So you took their antibodies and you absorbed them onto group O cells that were reacting, made an eluate back, and then you could test them against each other and any ABO group. And that's for anybody who is in a reference lab. It's a fantastic tool. In fact, I remember back when John Moulds first proposed to our staff that we spend a day or two making reagents in our reference lab by absorptions and elutions. So we would have all these antibodies against high instance antigens for all ABO groups. And it was quite an adventure, but it was fun. But sometimes it was kind of disappointing because they all didn't have titers like Mrs. Bigelow. So sometimes they wouldn't absorb and elute because they were very weak antibodies. So that could be a difficulty. So further complexities then arose in the LW story. There was Mrs. Wald in 1974. This was an inbred family composed of 3 LW negatives. G and B, remember they were the first two. Their antibodies were compatible with the cells of Wald, but the anti-LW of Mrs. Bigelow was incompatible. So now it shows that Mrs. Bigelow is different from the other antibodies. She stands alone by herself. Then they proposed the nomenclature of LW1 and LW2 and LW3. And about this time, back up a minute, about this time there was also studies of an Aborigine Rh null person in Australia, and this Aborigine also typed LW negative. So they felt that this was sort of the null of the LW blood group system. A very nice review was done in 1973 by Malcolm Beck at Kansas City, and he did it for the AABB annual seminar, is what they called it that year. And he combined notations of 2 different investigators, and he combined them to do LW1 through LW4, with LW4 being the Bigelow type. Now this is where the notations are very important, because if you have the serums labeled the LW1, 2 from the earlier years, and then from this other investigator you have different notations, and if you happen to have them in your collection of rare serums and cells like New York Blood Center does, LifeShare Blood Center got all of the rare cells from the Gamma collection, so they have all these and antibodies. Red Cross, the LA Red Cross, Irwin Memorial, well, I call it Irwin Memorial, but in San Francisco, all these reference labs have these very unusual samples. So it's very important that you know what those notations were and what they meant. And then in 1982, there was, Cystonin was the first person to report a new low instance antigen, NEA, and he reported that early on, and more studies were done after he reported it, and in 1981, they showed with some further studies that this NEA antigen was stronger on D positive cells than D negative. And then cord cells of either D type reacted equally well. So So in the literature, there is a paper on NEA, a new low incidence antigen, but then later on, there's another paper showing that it's really associated with the LW blood group system, or LW. So NEA has been designated as LWB, and it is the high incidence antigen is LWA, and that's the antigen of LW3 individuals, and then LWB is the low incidence antigen, or the allele. LWB, though, is high in the Baltic population, about 6% Latvian and Lithuanians, but apparently it is absent in the Asians and the Africans. So now you see an association with a particular blood group antigen that belongs to a certain population, and yet it's very, very low incidence in other populations. In 1996, there was another report, and it's the second example of allo-anti-LWAB. AK is the second example, and Bigelow, remember the lady who was unique and different, okay? AK and Bigelow are the same. AK cells are LWA negative, B negative with human antibodies and mouse monoclonal anti-LWAB. And as we go along, it's really fun to see these monoclonal antibodies being produced that have a certain specificity and can add to our knowledge of the blood group systems. And cells of AK's sister were compatible with her antibody. So these two, Bigelow and AK, are the only two that I know of. At least my literature search was ended earlier last year. I think I submitted my review sometime in September. So they're the only two that are allo-anti-LWAB. And again, having a sibling compatible is really nice. So it is nice to know. And you should not use these two antibodies to search for LWA negative donors, because they might call somebody who's LWA negative B positive to be LW positive, because their antibody detects the whole blood group LW antigens. So you need to use the antibodies that are known anti-LWA to search for LWA negative donors. And it's just important to remember all of these little things when you're working in a reference lab. What we used Bigelow's antibody for in the early years, it was so strong. That's another thing to remember, is that you can dilute that antibody. You dilute it and then you use it for screening. You don't use it neat because you'd waste up her. Maybe you only have a cc of her antibody. So you dilute it, and then when you get a negative, you go back and test it with the neat serum to see whether it's truly negative or LW weak. And it is important to confirm those with the neat serum. The nomenclature of the LW blood group system, and the International Society of Blood Transfusion established a nomenclature committee quite a few years ago, and I think we're up to 30-some blood group antigens. LW is the 16th blood group antigen, and these are the numerical notations for LWA. And you'll notice in LWAB and B, you'll notice that LWAB has a notation before LWB, and it's all because it was described before anti-LWB was. So normally you would think, oh, you should have an LWA, LWB, LWAB, but it was in the order of the discovery. Another thing is that LWA was always designated for the first antigen in the system to be recognized. Well, sometimes it can be a low incidence. So then, and are we confused or what? It's just like our English language is confusing, and when I tried to study Spanish, I mean, you had male and female and whatever else, so, you know, it can be a little bit confusing, but Anyway, I think with the nomenclature, though, that it's really nice, and they also probably are going to be doing away with these superscripts and subscripts. And the computers, like, for instance, they are naming things now with 3 capital letters or whatever to conform with the computer system. So those LW1 and LW4 designations that we had are obsolete now. You will not find them as far as nomenclature goes. So what are some of the characteristics about the LW antigen that help us serologically? And how many of you know about Dr. Larry Katz? You ought to know because he's in California. So you need to go Google Dr. Katz. And of course, George Garrity worked with Dr. Katz for many, many years on autoantibodies. And Dr. Don Branch also did his work here, and he's now in Canada. Well, in 1982, they developed they called it ZAP, I think. I call it Z-ZAP. It's a reagent that's composed of an enzyme papain and a chemical DTT. Well, workers in 1984 observed that anti-LW was negative with these ZAP-treated cells. What they discovered was that the component in it that destroyed or weakened the LW antigen was the DTT, not the papain, but the DTT. So the LWA, AB, and LWB are all negative with DTT-treated cells. So if you suspected an antibody to a high-incidence antigen, and it appeared to be stronger with D-positive cells than D-negative cells, and you wanted to see whether it was LW or a mixture of a high and anti-D, you would DTT treat the cells and retest them, and if it was anti-LW, everything would go away. If it was anti-D and LW, the D-negative cells would go negative, and you'd have the D-positive cells reacting. And then if there was anything else in it, you might be able to figure out a pattern. You do have to remember, though, that whenever you use these chemicals or enzymes, that there might be other antigens destroyed. And one of the first things that was described with DTT was that the KEL system antigens are destroyed with this, and YTA. So the Antigen Facts book is a good one to have if you don't have it, by Dr. Reed and Christine Lomas-Francis. Although I understand they're working on a 3rd edition and it should be out pretty soon. So fact book, all you ever wanted to know about blood group systems in a little pocket book. So it's kind of a neat thing to have. Christine Lomas-Francis and Dr. Patricia Tippett in 1985 showed that pronase also destroyed the LW antigen. Pronase is an interesting one. You can make it up and you can freeze it and use it. It does not destroy KEL system antigens. It doesn't destroy any of the rest of them. So in a way, you start out with the DTT, and then you could use the pronase if you needed to. Then Jeff Daniels in 1992 showed that 2 sulfhydryl reducing agents, DTT, of course we knew about that, and AET destroyed or reduced LWA and LWAB activity, showing a requirement for intact disulfide bonds. And we attempted to use AET in our laboratory to use this chemical as another tool. And unfortunately, one of the things we found that it tended to destroy the NOP system antigens too, or weaken them. And of course, NOP system antibodies are frequently very, very weak. And so oftentimes we didn't find AET as useful as we did the DTT and the pronase. LW human and monoclonal antibodies, allo anti-LWA and anti-LWAB sera of individuals who are genetically LWA negative B positive or LWA negative B negative respectively. Most are stimulated by transfusion, one was by pregnancy, and a male donor who was immunized to produce anti-D also produced anti-LW. Anti-LWB, there's examples in Finland, and there's one unreported case in Canada. There's also a monoclonal anti-LWAB 3 of them are IgG1. 1 of them is IgM. This also brings up another interesting point about the serology of LW. When we first started investigating different examples of anti-LW that were referred to our reference laboratory back in the 70s and 80s, we discovered that our reference lab, which most reference labs today too use the tube technique as their first technique, we discovered that some of these anti-LWs reacted at room temperature. And then you carried it through the 37 and indirect antiglobulin test, and they reacted there as well. But what it looked like at room temperature was back to, it looked like an anti-D-like antibody, okay? When you got to the antiglobulin test, all cells were reacting. So it was our clue when we saw that we probably had somebody who had anti-LW and not anti-D. And in particular, it was important because these were people who had never received D-positive blood, never been pregnant, and so there was no real reason for them to have anti-D in their serum. This is what we saw the most, probably, is transient suppression of LW antigens and production of anti-LW. And they got referred to us because they really thought they were dealing with genetic LWA negative, and they were really transient suppression. The first example was by Carolyn Giles and Lungard in 1967. They had a pregnant D-negative woman. Her cells were LW negative, but LW positive 12 months post-delivery, okay? So sometimes it took a while to show whether this was really transient or not, especially if you did not have a sibling that was compatible. In 1971, Dr. Bruce Chowan in Canada and co-workers had 3 cases of what appeared to be suppression of LW. They suggested that it was not anti-cell, but temporary LW negative with production of anti-LW. And we had quite a few examples of what we called anti-LW that were really not from a person who was genetically LWA negative. Perkins in 1977 had the first case of suppression and production of anti-LW in Hodgkin's disease. The patient's cells were LW negative and there was anti-LW in the serum. The patient donated a unit of blood 5 months after the antibody was discovered. They didn't test the unit and it was given as an autologous unit. 3 months later, the patient donated a new unit, and this time they did the typing on it and found that the unit typed LW positive and there was no anti-LW in the serum. So again, follow-up studies can really sometimes confirm your suspicions or actually give you more information. And then there's autoantibodies, and we had quite a few of these as well. And a lot of times autoantibodies were referred to us because people weren't able to, in their particular hospital, they didn't have the time or the reagents or whatever to do absorption studies and rule out autoantibodies. So the very first ones were seen back in 1967. And of course, we see that Dr. Levine was pretty much involved with a lot of these cases. They had 6 cases of autoimmune hemolytic anemia. What was important about this is that 5 of the 6 cases had other Rh-related autoantibodies. So autoantibodies can show single specificities. We know probably the most common is the autoantibodies that show a little e specificity. And you notice I didn't call it auto-anti-little e, I called it little e-like little e specificity. But other Rh ones were frequently found. And Voss in 1973 had autoimmune cases, and he had auto-anti-LW and concurrent Rh antibodies. And sometimes the Rh antibodies were actually alloantibodies. So there really certainly is this relationship between LW and Rh. And the molecular kind of shows why that is, as we'll get to in a minute. And then Jane Swanson again, and Jane has retired. She did go to the University of Minnesota and worked for some time, but I'll tell you, if you call Jane today, she goes like this. She talks and loves blood banking. She reads transfusion. She's very much keeping up with everything that goes on. And in fact, she's the one that had the cold case that they discussed, and it got published in Transfusion. I think it was last year on a Duffy anti-FYA that was unusual. She reported a patient with, in which there was donor-derived antibodies to LW, RH, and there was even an anti-M in this. It was a bone marrow transplant, chimera. And then in 1973, there was a study of 45,000 patients and donors. In that, they found 10 examples of cold reactive autoanti-LW. Cold reactive. Remember when I said about the cord cells having stronger D antigen? Well, guess what else cord cells have? Little I antigen, don't they? The cord cells are little I, and the big I hasn't become expressed on the cord cells yet. Through development it would be. So there are examples of cold reactive anti-little I's that really you have to distinguish whether you're dealing with anti-LW or anti-little I. But they can be a very, very useful tool. We used to be able to have them on the commercial reagent panels. Anybody in here old enough to remember the reagent red cell panels that had cord cell as number 11? And it was a, oh, they're great, good. But then we had to quit, you know, rules and regulations. We didn't have permission to use these cord cells, and hospitals were getting in trouble sending them to us, and we were selling them, you know, as part of a panel. So, but we still kept getting cord cells, and we actually washed up good low cord cells, and we had liquid nitrogen, and we froze our cord cells away so we could still continue to use them on studies. But anyway, there are cold reactive anti-LWs. Clinical significance. Mrs. G had 13 pregnancies, no anti-LW until she got transfused. BW had allo-anti-LWA. She had 8 children, and there was no HDN. Bigelow, anti-LWAB, remember her real strong titer? Her third baby had a weak positive DHE, but there was minimal HDN. I call that serological HDN versus clinical. So there's not been reports of clinical HDN with anti-LW. Transfusion data, LW positive to patients with apparent allo-anti-LW with red cells transiently LWA neg or LWA neg B neg. So most of the data in the literature is on the transient ones. The ones before that I mentioned were about the only ones on the clinical side for genetic ones. Mike Gallus in 1977 had a 14-year-old male patient with anti-LW. They gave 50 units of D negative LW positive blood and no untoward effects. And most people who even have these transient or autoantibodies will tend to sometimes try to give RH negative blood, RHD negative, feeling that it survives longer, clinically possibly, that it makes us feel better because it's not as incompatible as the D positive blood. But people certainly could give, and you have to monitor each patient, as we know, and see whether it's possible to give D positive. As some people would argue that we are maybe wasting D negative blood for these patients when they might not really need it. So you evaluate each one of the patients as you find them. Cummings, again, reported in 1984 on an apparent allo-anti-LW. Again, there was no compatible sibling. And chromium survivals were done back then, and the MPA was low significance for this antibody. And they also, in that year, there were 2 other reports with the same results. However, in 1986, there was a report, in contrast, where there was an IgG3 anti-LWAB in an 85-year-old male with myelodysplastic anemia, and they radio-labeled LW-positive red cells, and there was evidence of destruction in this case. Here are a couple of other examples. In 1987, they gave Rh-negative LW-positive blood. The report in 1995 was the one that I found on MMAs, and 54.9% with D positive and 23.2% with D negative, and 5% is considered, 5% and lower is considered normal survival. So this particular antibody appeared to be definitely clinically significant, and the patient, even though it was probably transient suppression, this person probably should receive LWA negative blood. Cystonin again he's the one that had the anti-LWB patients, and they received blood with no reaction. One patient though, the estimated half-life of 2 ml of cells that they gave was 2.5 hours. So there is one patient that there's possible clinical significance. And now to the molecular LW, which explains a lot of the serology and everything that we had gotten in the past. And I have to tell you that I struggled with this part of the review because I'm not a molecular person, even though I think it's fascinating all the things that it can do for us. It has explained a lot of things and brought up some interesting things too. In 1983, Moore did studies and showed that it was a 40,000 Dalton glycoprotein. Mallison in 1986 confirmed it using monoclonal antibodies. And then Loy and co-workers in 89 showed that LW and RH proteins are part of an RH cluster, is what they called it. And then in 1990, though, they showed that RHD and LW polypeptides are different proteins. And they probably did the most fascinating work to initially show that they were different. Then Bailey in 1994 and 1995 published the work that everybody in the molecular field was really excited about. The LW protein sequence was similar to ICAM 1, 2, and 3, intracellular adhesion molecules. And then they described that LW protein, they named it ICAM4. So it's part of this group. And it really was a big breakthrough in the molecular field of LW. And then they looked at LW and disease in association with it, a lot with the molecular. And Parsons in 1999 showed that the Lutheran glycoprotein and the LW glycoprotein are members of the immunoglobulin superfamily, or IGSF. And then in Spring and Associates in 2000 and 2001 designated that LW group glycoprotein be designated ICAM4. So in a lot of the literature that you'll see in the molecular, they will refer to ICAM4, and they might put in parentheses LW, or they might not. So one of our participants in this seminar actually knew about LW or ICAM4, and I was so excited. I thought, this person's going to be a molecular person. I can tell right now. But anyway, ICAM4 now, they have done some work, and there's adhesive interactions in sickle cells. So now it becomes, I never would have believed that the LW blood group system and glycoprotein would be associated with the sickle cell disease. And actually, when I was writing this review, I saw this one paper, and so I emailed Dr. Tellin, and back came an email and said, oh, you have to have these publications, and it's really exciting news. And in 2006, she sent me a copy of paper that they had written, that they showed that LW red cell adhesion, hematopoiesis, and vascular disorders were associated, sickle cell disease, pathophysiological role for LW in human disease. LW contributes to red cell adhesion to endothelial cells and development of vaso-occlusion. And there were two other reports that I found in the literature, and there may have been some sense. This is 2012, but these were the ones that I found before I had to submit my review to immunohematology. In 2008, phagocytosis of senescent red cells is in part ICAM4 beta 2 integrin dependent. And then in 2010, these workers summarized the previous reports on molecular processes occurring within erythroblastic islets. So this particular paper is a really good one to read about it if you are interested in the molecular part of LW. And by the way, the review that I had has all of this information in it, all of the references. So you just need to go to my review and you'll be able to get it. So, in summary, LW and RH are associated. LW is absent on RH null cells that lack all the RH antigens. LW and RH are part of the immunoglobulin superfamily, IGSF. But LW and RH are two distinct proteins, and they arise from unrelated genes on separate chromosomes. The LW blood group protein, ICAM4, is a member of the IGSF subfamily known as intracellular adhesion molecules. The role of LW or ICAM4 in red cell adhesion contributing to vaso-occlusion in sickle cell disease and other vascular events. And by the way, Dr. Tellin, I know, is continuing her research on that, and so you and other workers. So probably if you did a search of the literature under ICAM4, you'd probably be able to see if there was anything more recent than those publications. And I want to just kind of summarize, and again, in my references, in the article I wrote, are these different reviews, but Beck was the one that did the first one in 1973. There's a lovely review by Gail Stilwell. She was a student at the time, and it was her paper for her certification in 1979, and it's in the Canadian bulletin, and it is referenced, I believe, in when you look it up in PubMed. And it's fascinating to read she gave the story, probably one of the first stories, real good, of the patients that produce these antibodies. And then in 1980, just a year later, Carolyn Giles in England did a very nice report in Immunological Communications. And Carolyn Giles is still alive today, retired. She's a fascinating lady too, because these people in the early years had other interests as well. In fact, her real interest and love was in Cheeto and Rogers. But her boss asked her one time to do a review of LW, and she really wasn't too interested in doing it, but they'd had patients. So she did this review of LW. And I can tell you that if you've ever done one, it is a labor of love, but you got to love the whatever you're writing on to do it. And then in Jill, it was 12 years later, Jill's story was at the New York Blood Center, and the American Red Cross Immunohematology Journal, she published a review of LW, and so that was the one. But can you believe that it was 2011 that is when my review, so I thought they want another review until I realized how much had come into play since Jill wrote hers, because there was very little known at that time about the molecular of LW. So Oh, and it's also confusing too. In the early years, I thought about hyphenating my name and using my maiden name, but Grandstaff's kind of long. So anyway, but I just decided, you know, my mom and dad put me through college and everything, so I ought to use this Grandstaff name. But if you go to look up molds, you might not find this journal article. It might be under Grandstaff Moulds, so another confusing thing. But, and there's an error on this. It should be pages 136 to 142. I didn't catch that until I pulled the journal the other day. How many of you get the Immunohematology journal? Now, students get it free, okay? And they started, American Red Cross started this journal, I don't know, a couple of years ago, and they're covering all the blood group systems. Now, here they're online too. I don't know how many are online or whatever, but I don't think this one's online yet. But this issue has my article in it, the LW one. It also has several other very interesting articles, and it was dedicated to John Mould. So there's a lot of fun stuff in there, and if anybody wants to look at it, I have this journal so you can get a look at it. But you might see if you can find out from the Red Cross whether you can get back journals, if you want it online, or I think it'll be online pretty soon, the back issues of Immunohematology are. So remember, though, the names, and you can go away today and say, well, I've seen the pictures of some of them, Jane Swanson, you know, and Dr. Levine, and I think these names are very, very important, and the history behind the blood groups. And then John used to say, remember the history. So anyway, we need to keep remembering it. We need to bring up the stories, and maybe someday, instead of writing these journal articles, I might write a history of blood groups and the stories behind them and the people behind them. I don't know whether I'll have it a sexy novel or what. We'll see. I might get some criticism of that. But the stories that we that have been in the field for so long can tell. I mean, one of the things about it at the Gamma Reference Lab, we had these and Nancy will remember this, we had these family studies, and we needed to get some family samples from family members, and some of them were in Louisiana, Kentucky, you know, the boondocks, and so we called on our local sales rep, and Jimmy Turner was a rep in one of the areas, and so, Jimmy, we need these samples, here's the names of the people, and okay, I'll go draw them, you know, he got his kit from the local hospital. Can you see us doing that today now? Probably not. But anyway, Jimmy went, and the stories that he had to tell about staying in this fishing cabin that leaked and everything, and the stories about the family and that. And so it really is something to know the history behind this. And if you read those reviews, there's quite a bit of stories in them about the different families. So anyway, I've shared today with you about LW, blood group, and maybe I've stimulated you a bit to be interested in the blood group systems. And Marion Reed does have a new book out. It's wonderful. Oh, and you go on PubMed and you look for blood group antigens and antibodies, and there's a great whole thing on, I don't know who wrote it, on PubMed. There's a few errors in it. You might see if you can find them, but there's a lot of interesting and fascinating stuff out there. I thank you for your attention.